Thank you very much, Dr. Arner. And then we'll move to the next presentation by Dr. Vidal Puig. Tony has done wonderful work at the Cambridge Metabolic Research Laboratories in the UK, where he has been for many, many years now, no? And his particular field of interest has been the molecular mechanisms of energy balance. He has done a lot of work on energy expenditure, on lipotoxicity, the adipogenesis, and he's going to present today on the brown adipose tissue as an antiobesogenic and antidiabetic strategy. So thank you again. Thank you also for coming here, Tony.
Well, it's always a pleasure coming to Barcelona and meet all my friends and colleagues. And thanks for the invitation to talk about brown fat. I think I will start by presenting a bit our view of the problem. For us, we have an adipocentric point of view of the metabolic syndrome. We think that although you can get to the metabolic syndrome probably through different entrances like muscle or the liver or the hypothalamus, the most common entrance is through obesity. And for us, the key problem is not how obese you are, but actually the mismatch between the amount of fat that you need to store and the capacity that you have to store. And the key word for us is lipotoxicity, which we define as the topic accumulation of nutrients in the form of fat in organs that are different from the white adipose tissue, organs that are not designed to be able to store fat. And then is the conflict between this type of toxic event and the genetic makeup of the specific organs, which determines what is the specific phenotype that the individuals develop. Not everyone with metabolic syndrome develops everything, and we conceive that is this type of interaction between the toxicity of the nutrients and the genetic vulnerability what determines the phenotype. The way how we approach the problem of lipotoxicity is from three different themes. The first one is very close to what Peter has been mentioning, which is the functionality of the adipose tissue. So we think that if we improve the functionality of the adipose tissue, we make the adipose tissue more able to store fat and flexibly release fat when the other organs need it, then that could prevent lipotoxicity in other organs that will contribute to maintain energy homeostasis. The second thing that is relevant for today is the concept that if your adipose tissue fails and then there's lipotoxicity everywhere, the second strategy that you may have to try to prevent the toxic effects of the nutrients is by burning them. And over the years, we have followed different strategies. We were looking at exercise, trying to mimic the effects of exercise through increasing the amount of mitochondria. We were trying to make the mitochondria less efficient through uncoupling. And most recently, we have been focused in trying to make the white adipose tissue more brown. And the brown adipose tissue as an organ that can contribute to become less efficient and increasing the transformation of nutrients into heat. The third approach is about qualitative aspect of lipids. We think that not all the lipids are the same. We think that there are lipids that are more toxic than others. And we think if we understand this, how the different lipid networks in different organs work, how these lipid networks in different organs evolve during the process of the natural history of diabetes, then we can potentially divert nutrients from the most toxic pathways to the less toxic pathways, and that may contribute to prevent or ameliorate the development of the problem. From the point of view of the adipose tissue, what I'm going to present to you is data from the two key molecules that my lab is working on. One is VMP8, which is a modulator that activates the brown fat, but also will present data from LR11, which is the extinguisher. You need to consider, I would say, most of the people, they are interested in the brown fat because they want to activate the brown fat, because they want to lose weight because they activate the brown fat. However, there is a problem. If you activate the brown fat and you make too much heat, you can get burned. So you need to have systems that can block the excessive production of heat. And it's the balance between these two systems that can maintain the production to serve the purpose of maintaining temperature. So from the point of view of the adipose tissue, then what we have is the white adipose tissue, the brown adipose tissue, and there's something in between, which is what is named the bright or beige adipose tissue. The bright or beige adipose tissue exhibits some of these brown fat cells interspersed within the white adipose tissue. And it is relevant from the point of view that human beings are more likely to have, it's been suggested, to have more of this bright adipose tissue, in this respect being more likely to be useful for therapeutic purposes. But this is totally debatable. What we know from animal models is that if you have an animal model that is prone to develop to obesity, if you raise them at higher temperatures, the animals, the animal becomes obese. If you have an animal that is lean, even you raise at higher temperatures, these animals become more obese compared to animals raised at lower temperatures. And this diversion between obese and lean occurs as soon as you put them in cold exposure. The second thing that we know is that the brown fat in animals not only makes heat, but actually can help you to control the amount of lipids that you have in plasma. Because the brown adipose tissue, what it will do will be to suck it up the lipids in order to burn them. And then you have this type of data coming from animals that suggests that when they are exposed to cold exposure, you may have, even after gavage of a high dose of olive oil, you can have the excursion of the triglyceride in plasma to be blunted. So these offer opportunities for the possibility that activating the brown fat will not only help you to lose weight, but actually to ameliorate your metabolic parameters. And this is an example that is being done with respect to the activation of the brown fat in the cold. My image of the brown fat is like a vacuum cleaner. You basically activate the brown fat and you clean up your muscle, you clean up your liver, and you clean up your blood. Because the brown adipose tissue will suck it up, all these lipids, in order to burn. So you could potentially take advantage of this to ameliorate some of the other manifestations of the metabolic syndrome. In summary, I think some of the potential advantages derived from brown fat activation increase, obviously the increase in energy dissipation that can contribute to control body weight. An important aspect of it is that this doesn't seem to have cardiovascular effects. And this is important in the context of previous drugs that were activating uncoupling of the mitochondria, but they were non-selectively active uncoupling the mitochondria from the heart. So this strategy uses the same approach, but preventing the complications of having heart problems. On top of this, you may have a potential improvement in dyslipidemia and atherosclerosis, an improvement in insulin resistance, and improvement in fatty liver. And also, maybe there are some suggestions that by depleting this type of excess of nutrients, you may not prevent cancer, but you may delay the development of cancer. You may delay the progression of this, and also may have some impact in neurodegeneration, that some aspects of the connection between obesity and neurodegeneration in the context of lipotoxicity may be ameliorated simply by removing the substrate from this. The last two is, I will encourage you work in some of these fields to pursue this further, but that may offer some opportunities there. Peter will have said for many years, forget about this because this is a mouse thing or whatever, we don't want to deal with that. Well, bad luck. Now it seems that human beings can have brown fat. Not all of them, but in theory you can find some of these. This will correspond to the canonical brown fat, and also the possibility of developing some of this beige brown fat. And then this confirmation that what appears to be brown fat in humans is brown fat because they have immunohistochemistry for the uncoupling protein 1. And also we know that, however, there is a problem. And the problem is the people who we need the most the brown fat, we are the people who have the less brown fat. The older you get, the less brown fat you have. If you are obese, then you get less brown fat, probably because you are thermoinsulated and you don't need to make heat. And you have diabetes, you may have less brown fat, which we don't know if it's cause or consequence of the diabetes. What we know, which is very interesting, is that if you take some of these individuals and you put them in the cold, they can rescue the amount of brown fat that they can exhibit. So which suggests they don't have the brown fat, but they may have some of the precursors still there, waiting to be activated or to promote the differentiation and activation. And that offers some possibilities. So the challenges of making brown fat therapeutically viable for humans is two ways. You need to have enough of this brown fat, which is a problem. And second, you need to be able to activate this brown fat. So from this point of view, the strategies that we are following is to try to make more of this brown fat. We need to understand how we can differentiate brown fat cells from stem cell differentiation. And on the other hand, given that the brown fat is activated in the context of the sympathetic nervous system, but also in because the activation of the sympathetic nervous system by itself can create a lot of problems. You need to find a way how to make this brown fat more sensitive to the stimulus of the sympathetic nervous system or other alternatives to prevent some of the side effects. I will try to explain this a little more clearly. So when we have been in the clinic trying to reverse obesity, what we know is that approaches that increase energy dissipation work to help you to lose weight. There's no doubt about it. The problem is that the drugs that were directed to increase energy dissipation also increase the sympathetic tone to the vascular wall, to the kidney, and to the heart. And that resulted in inappropriate cardiovascular side effects that were not acceptable despite the fact of losing weight. However, what that means is not that thermogenesis is not a good approach. It's that you need to find a way how to selectively or predominantly activate the dissipation capacity without trying to affect the sympathetic innervation in other places. From this point of view, our key concept from the brown adipose tissue is that it's like a vacuum cleaner that can eliminate lipotoxicity in peripheral organs. And we know that this thermogenesis depends on the activation of the sympathetic nervous system. So I think probably most of you are familiarized with this, but basically the system as it works is that norepinephrine activates the beta-3 adrenergic receptor. Then there is a cascade that goes through the pKa phosphorylation. And then you have two responses. One response is the acute response. Basically, you activate lipolysis, you facilitate the release of fatty acids. These fatty acids go to the mitochondria, and then the mitochondria and the brown fat are able to make heat. However, if the plan is that you're going to be needing thermogenesis for the long run, there's another adaptation that makes you to have more mitochondria and more cells, brown fat cells, that increase the capacity of the system to make more heat. From this point of view, what we want trying to do is to increase the capacity of the brown fat cells to make more of them, but also to be activated more fully. And then the opportunity is coming from the observation that brown fat activation does not only depend on the sympathetic nervous system. I mean, over the recent years, what has happened is that there's been some evidence that peptides coming from the heart are able to activate brown fat. That FGF21 and Frances Villarroya has done a beautiful work on this area, is able also to activate brown fat and act on the brown fat itself. People in Boston is suggesting that another muscle-related type of hormone that is irisin that may contribute to this. This is controversial and is totally improved. In our case, what we found is about BMP8, BMP8B, which is a bone morphogenic proteins. When you think about bone morphogenic proteins, they'll get misled. It's not about bone. It's just the name. It could be named a different thing. But the bone morphogenic proteins are members of the TGF-beta superfamily. They are secreted proteins that bind type 1 or type 2 transmembrane receptors to regulate transcription via the SMAD pathways. And what we learned about BMP8 was the following. I would like you to follow some of the data. As many of these genes are expressed in the testes, but BMP8 is highly expressed in the brown fat and is highly expressed in the brain. And this is very different from all the different organs. So BMP8 is in the right place to control thermogenesis. The second data is physiological. I mean, if you get high-fat diet or cold exposure, thermogenesis should be activated. And what you find is that BMP8 is the only BMP that is highly increased during high-fat diet and super-induced during cold exposure. For BMP7 and BMP4, that may also contribute partially, there's an induction with the cold exposure, but nothing compared to what is BMP8B. However, this data for me is one of the most important ones. BMPs are morphogens. You're supposed to have them in precursors. They may contribute to the differentiation, but that didn't happen for BMP8. BMP8 appeared in what is the mature form of the adipocytes. It's one of the BMPs that appear, not in the process of cells that need to be differentiated, but actually in cells that are fully functional. And that's an important difference. So when you work with the knockout mouse, one thing that is very important, I would recommend you to focus when you are in your own animal facilities, is that when these animals are being raised at 20 degrees, we lose substantial knockouts. It's only 11%. This is well below the Mendelian ratio. However, when we raise the temperature of our animal house, what we get is restoration of this, and by taking them to 25, we restore normal which is telling you that the exposure to the cold that happens after the delivery is challenging these animals and is compatible with losing these animals during this process. Since then, and given that we work with a lot of animals of thermogenesis, we raise all our colonies at almost at thermoneutrality. So these animals, either on chow diet or in high-fat diet, they become obese. There's another detail that is not in this slide, is that these animals eat less. I think my friend Javier always said that if you are obese, probably it's because you eat too much, but this mouse can get obese and it eats less. And when you have one of these examples, that means that these animals actually spend much, much less than they are supposed to spend. This is an experiment that basically, I'm not going to enter in the detail because it's beyond the scope of this presentation, but basically looks at the maximum thermogenic capacity of the animal. It's an experiment that you need to do by having these animals at 4 degrees, build up all the brown fat that they can make, hit them with norepinephrine, and look for the difference. And when you see this is in these animal models, their maximal capacity that they can dissipate is diminished. It's probably the most clear observation that not having BMP8 prevents this thermogenesis from happening. And also because the first part, which is required for the activation of thermogenesis, the lipolysis, what these data show is that having BMP8 in the system, what it does, amongst other things, is to facilitate the lipolysis, releasing the fatty acids that are going to be uncoupled. So in this respect, BMP8 is not able to promote thermogenesis, but it's able to amplify the signal of the sympathetic nervous system that with similar type of input, you get much more response in this sympathetic, in the thermogenesis response. We can deal with this in the questions because there are some issues with respect to selective innervation of the sympathetic nervous system that are very relevant here. So in some way, what we learned from this experiment is that in the BMP8 knockout mouse, there's an increased sympathetic nervous system activation as compensatory of a blockade that occurs below the MAP kinase 3 and 6. So these animals, if you look at the sympathetic innervation, is very high. But this is the result of having a system that doesn't allow the signal to go through. The thing that we have learned now from these animals is that if you overexpress BMP8 in the adipose tissue, there are two things that we may get. It's also an increased vascularization and an increased innervation, which suggests that they may also contribute to the aspects that are essential for the brown fat activation, which is to have enough innervation there to be able to activate that. And this is an effect that happens not in the acute way, as we observe with the BMP8, but also to the medium or long term. So I think our next paper will be related to the role of BMP8, chronic activation of BMP8 promoting innervation and vascularization in the brown fat as factors that contribute to increased thermogenesis and also in white adipose tissue to promote beiging. So what we have seen here is a system that helps you to improve the activation of your brown fat system. So for the same signal of sympathetic nervous system, you amplify the response. However, we think, and we have observed data that suggests that when there's an excess of activation of brown fat or an excess of the sympathetic nervous system, there's a feedback regulatory loop that tends to shut down the system. And we identify LR11 as a regulatory, as a negative regulator of brown fat. So what is LR11? LR11 is a member of the LDL receptor family. These are big molecules, so we don't like them because they are too big and very difficult to study and with multiple functions. So these type of molecules contribute to the uptake of plasma lipoproteins. They bind ApoB and ApoE. And they have 1 to 4 clusters of ligand-binding repeats that we think are related to lipophilic molecules. We also know that they are highly expressed in the brain, where they are regulated by omega-3 fatty acids, which has something to do also with the fact that these animals in the long run, also develop neurodegenerative diseases. What we learned is that on top of all these locations, LR11 is also located in the white and the brown fat at a very high level, and this was not known. So the way how it works, at least in the brain, is this LR11 is a type 1 transmembrane receptor. You have an extracellular component, an intracellular component, and intramembrane component. There are some molecules that are shedded that will separate the extracellular part of it, becoming a soluble molecule that can signal at a distance. But also, when that happens, there's also the release of the cytosolic form that goes to the nucleus and can activate some aspects of the transcription. So what we are going to study is a total knockout of LR11 that basically decreases the amount of the soluble part, but also decreases the capacity that the intracellular has on the signaling or the transcriptional aspects of it. So what we learned from this animal is that if you don't have LR11, the animal is leaner. It's not shorter than in the picture. That's a bad picture, but the animal is the same size and basically is leaner. So the animals have less white adipose tissue in different depots, subcutaneous, epididymal, mesenteric, and retroperitoneal. This is the brown fat. And some people, when they look at brown fat, they always look at the body weight, at the weight of the brown fat, which is wrong, because obviously what you see here is that this is more heavy than this type of brown fat, but this is at the expense of more white fat going around. But what you observe there is that these animals have a more brown fat with less fat inside, which suggests that the animal is burning more, and that's why you see less. And more importantly, is that when you put them on a high-fat diet, these animals actually are protected from the development of fatty liver, in suggesting that the brown fat may be sagging up and cleaning it up. What you know from these animals is that, okay, this is a wild type and a normal diet and a high-fat diet, but these animals are resistant to high-fat diet. You can feed them with a high-fat diet and they cannot put on weight. And what we learned from this analysis of covariance of the body weight and energy expenditure is that these animals, in fact, are spending more energy. So not having LR11 results in an increased expenditure of energy, so having LR11 should be interpreted as protecting from energy expenditure. So the other thing that we learned is that also the white adipose tissue of these animals have increased expression of UCP1, BMP-AB, that's very relevant here, PC1 alpha, and other markers that suggest, as well as elongase 3, that are suggestive of browning of the white adipose tissue. But I will not say that the browning of the adipose tissue is the important thing in this animal, because in fact, what we use is the white adipose tissue as a marker of, for example, 60% here may not be that relevant. 60% of almost nothing is almost nothing. However, the 10% that may be increasing the brown, canonical brown fat, we think is much more important in absolute terms to the contribution to the phenotype. However, for us, this information about the sympathetic activation goes to the white adipose tissue, and this is a reported system of the activation of the sympathetic tone. This is the opposite experiment that we saw before. This is the maximum tryptogenic capacity of the animals when you inject with norepinephrine. But this experiment is very different because we don't expose them to cold. We put them at thermoneutrality, which means we decrease to the maximum the capacity of the brown fat, and we assume that the differences in energy capacity are coming from the high-fat diet. So we are testing high-fat diet-induced thermogenesis in the system. And what we observe is that in the LR11 knockout mouse, even a high-fat diet and at thermoneutrality, the lack of LR11 allows the system to burn more energy. Okay? So that suggests that LR11 may be more relevant even in the diet-induced thermogenesis than in the cold exposure. More data you have here. This is the effect that we observe in wild-type adipocytes and knockout adipocytes when we inject with norepinephrine. This is the normal response in white fat, in white adipocytes with oxygen consumption being increased by norepinephrine. But in the knockout mouse, when you treat them with norepinephrine, the response is much more stronger. And here we have the opposite type of experiment. All of them are control animals. Here is control animal with norepinephrine increases the amount of oxygen consumption. Here, basically what we do is we incubate with the soluble form of the LR11, the one that was cleaved. And we can prevent the effect of norepinephrine by blocking the activation of the brown fat and oxygen consumption in the system. Other things that we can see from here is that whereas BMP7 in this case can increase UCP1, PC1 alpha, and CDA, this response is much more evident in the knockout mouse. So the knockout mouse, when you have this type of activators, the response is much larger. However, this response is totally blunted when you incubate with the soluble form of LR11. LR11 can block even the facilitation of BMP7 in the system. And this happens for the expression of UCP1, PC1 alpha, or CDA. And this is with respect to the signaling. Again, we have a similar paradigm. BMP7 can increase this phosphorus MAT 1, 5, and 8. LR11 can block this effect. In the knockout cells, BMP7 can have a much more activation of these MATs, and this still can be blocked by LR11. So that means these two systems are talking to each other with respect to activation and blockade. These data are important for interpretation. It's a simplistic interpretation that says, well, this thing blocks, and under conditions where you need to block, that's when the system is going to be activated. However, if you look at the physiology that happens in mice, what we observe is that LR11 is higher under conditions of cold exposure. So LR11 is higher when you need to make heat. So you have a break being induced when you're making the heat. So the interpretation is not that LR11 helps you not to spend energy, but that LR11 comes in when the system is overactive, trying to prevent the system getting out of control. For us, this is very important because we observe that many of the genes, many of the programs that we see induced in some of our differentiation programs are not directed to facilitate the progression, but actually to impose a break that facilitates the very strict organization of the process. And your interpretation, when you interpret your levels of LR11, should be in this direction. And this, for example, also happened with respect to the role of LR11 in the concept of humans with respect to the BMI and the weight loss. What you have is that the fatter that you are, these individuals have more LR11, and also that during the weight loss, what you have is you lose weight and your levels of LR11 decreases. So I think the interpretation of this should be more on the concept of break than on the concept of facilitating. So from this point of view, we think LR11 is a negative regulator of thermogenesis. We think that this prevents from the system getting overheated, and we think that if left unchecked, this may increase, or this will lead to an increase in the recruitment of the brown fat cells. Some of the conclusions here are not relevant to the whole thing because it's a whole program, but I will concentrate on the third one, which is that brown adipose tissue activation can eliminate the excess of nutrients and improve brown adipose tissue. And the only thing I would like to say with respect to LR11 is that, okay, we work in obesity and we want to get people to lose weight by increasing thermogenesis. But there are other conditions where preventing thermogenesis may be very important, states where you need to save energy. And I'm thinking about cancer, I'm thinking about people who are burned, where we know that the system is totally activated to increase brown fat and energy dissipation, or even in conditions like anorexia nervosa, where you may want to prevent these individuals from expending unnecessarily energy. So in this respect, the focus in the brown fat, from my point of view, should go not only for the energy balance, but also in the context of saving energy in diseases where this is. Last few slides to tell you where we are going now. I have said we were looking for activators and controllers of the activation of the brown fat. However, if obese people, we don't have brown fat, we cannot get any treatment that will activate our brown fat because we don't have brown fat. So we need to make more brown fat. However, the observation that people who are obese can develop brown fat when exposed to the cold tells you that these individuals might have some precursors midway through the process somewhere that can be activated under the proper stimulus. So the whole point about this is what we are trying to do is to work with pluripotent stem cells, human pluripotent cells, to try to develop two protocols. One is from the white adipose tissue that we have optimized now that could help us to understand not only how the white adipose tissue or white adipose has been made, but actually how can you make more of the browning. So it's a tool that we can use for manipulation. But most importantly is the concept that we can take, for example, these pluripotent stem cells and try to differentiate them into brown fat cells. Our strategy is not to hit them with a specific drug that will give you a brown fat cell, but actually the strategy is to mimic the specific stages through which these pluripotent stem cells follow until they become a brown fat cell. And our interest is not here. We are not interested in this. We are interested in the intermediate stages, because if we can grow up enough of these intermediate stages, and we can recognize some of the proteins that are covering the membranes of these intermediate stages, we may potentially develop some drugs that may take these cells and advance their stage toward final differentiation of the brown fat. So which means we will not need to have any transplantation strategy. We will not need to do anything really weird, but actually rescue the capacity of an individual to make their own heat. So that's what we are trying to do these days with support with the ERC, which I think is very helpful. And also, these cells are becoming now experimental tools that we are collaborating now with people from genetics, where we are getting some of these variants that are related to the context of obesity, and we are looking, for example, with Torben Soren about people from Greenland to look for brown fat, or people looking also in the context of India and different types of adipose tissue biology in India, to try to recapitulate that in these systems and trying to define how they may affect the development of the adipocyte in the context that Peter has been brought out, which is the dysfunction of the adipose tissue in the context of the metabolic syndrome. So this is our team. Maybe the picture is a bit old because they look younger than they are now, but we don't have a lot of replacement in the group. So all of them, these are our collaborators, our funding bodies, and so on and so on. And I think we are getting to the, if this company wants to sponsor a football team in Cambridge, we are open for sponsorship for the next season. So at the present moment, my lab is sponsoring this team, but these guys are growing, and then we need to replace the equipment. So I think any sponsorship from the company will be more than welcome. So with this, I think I will take any questions. Thank you.